Hello, my name is John Brown. I'm a second year student here at UCSD studying biochemistry, and my project is on the study of chaperone activity of insulin degrading enzyme using amyloid beta and HIV-1p6. Insulin degrading enzyme, or IDE, is a ubiquitous zinc metalloprotease that was originally hypothesized to be involved in insulin catabolism. However, it was later discovered that this enzyme degrades a whole range of amyloidogenic substrates, including insulin, amyloid beta, amylin, glucagon, and many others. This figure here represents the crystal structure of IDE and its substrates. IDE is made up of four alpha-beta role domains. Together, these form the N-terminal domain and the C-terminal domain. The catalytic site is located at the N-terminal domain, and the C-terminal domain is responsible for substrate recognition and dimerization. The figure on screen represents IDE in its monomeric form and is around 120 kilodaltons, while the dimer is approximately 240 kilodaltons. The peptide substrates are shown here in red, and this circle represents where those peptide substrates would bind to on IDE. Where the substrate binds, it forms a beta strand in the catalytic site of the enzyme. Therefore, IDE likes to cut monomeric amyloidogenic polypeptides. And this is how IDE is involved in proteostasis. As mentioned, one of IDE substrates includes amyloid beta. Uh, Amyloid beta monomers aggregate to form toxic soluble oligomers, which seed the formation of larger A beta fibrils and eventually the formation of plaques that are commonly found in the brains of those with Alzheimer's disease. It is hypothesized that the toxicity of the soluble oligomers of A beta induce neuronal damage that leads to the onset of Alzheimer's disease, and that the amyloid plaques are the body's defense mechanism to confine these toxic oligomers. While IDE degrades A beta monomers to help prevent the formation of toxic oligomers, it also acts as a dead end chaperone. The role of chaperone proteins in proteostasis is to prevent aggregates of misfolded or aggregation-prone proteins. The green arrows in this figure represent the processes promoted by the chaperones, which lead to stable protein conformations, while the red arrows represent the processes that are inhibited by chaperones, preventing the formation of misfolded proteins and protein aggregates. IDE similarly prevents aggregation of A beta by binding to it and preventing its participation in the aggregation pathway. Recent reports indicate that IDE degrades an HIV peptide known as P6. This is postulated to interfere in IDE-mediated insulin processing, as well as A beta clearance in individuals with high viral loads. In our lab, we seek to understand the molecular basis of this chaperone activity. To do this, we work with two different substrates, A beta, which is amyloidogenic, and P6, which is completely soluble. By comparing IDE's interactions with these two substrates, we seek to understand the mechanism of chaperone activity. Here's some experimental data that I collected. A beta 42 and A beta 40 are the two major isoforms of amyloid beta, and I measured their aggregation using a thioflavin T assay. Thioflavin T is a classic amyloid-specific dye that fluoresces when it binds to amyloid fibrils. As we can see for A beta 42 and A beta 40, when they are by themselves, they aggregate very rapidly to create this classical sigmoidal profile as shown in blue. In the presence of catalytically active IDE, we see no aggregation for either of the peptides as shown here in green. In the presence of catalytically inactive IDE, more importantly, we do not see aggregation for either of these peptides. And this is indicating that inactive IDE can indeed act as a chaperone protein. Inactive IDE was prepared with a point mutation, and EDTA was added to ensure that its proteolytic function was inhibited. And this is because IDE is a zinc metalloprotease. I analyzed the aggregation assay I showed you earlier by running an SDS page. I then stained this gel using a silver stain. In this column, we can see the control, the A beta 42 by itself. After aggregation, we see that there is no band density here. In the presence of active IDE, We see that there's no band density of A beta 42, and this is because the active IDE has hydrolyzed the A beta 42 monomers. In the presence of inactive IDE, however, we see that the monomeric band of A beta 42 is still present post-incubation, thus indicating that it has performed its chaperone role without hydrolyzing those A beta 42 monomers. We can monitor degradation of A beta and P6 by IDE using NMR spectroscopy and calculate the rate at which both substrates are cleaved by the IDE. Note that in order to prevent A beta aggregation, as we saw in the previous slides, all NMR experiments were done at 2 degrees Celsius. A beta 42 is cleaved by IDE at 2 locations, here and here, while P6 is cut at 1 location, right here. The corresponding hydrolysis rates are similar between 30 and 100 picomole per minute at 2 degrees Celsius. Our lab just recently submitted a paper which explains the molecular basis of this chaperone activity. In the case of A beta 42, most of the time it forms this dead end kinetic trap, but can also form this sparsely populated complex, which is primed for proteolysis. In the case of soluble P6, it can only form this productive complex, which an active enzyme can proteolyze. This is all very exciting information, and we are actively trying to understand this phenomenon better. I would like to thank my PI and mentor, Dr. Lalit Deshmukh, for guiding me throughout this project. I'd also like to thank Spencer Nelson, who was a graduate student I worked with closely during the duration of this project. I'd also like to thank Dr. Tyler Rogers for supporting my research, as well as the TRELS program for giving me this wonderful opportunity. Thank you.